I would like now to go on with another speaker. Uh, he is Mr. GERLI, and he will present us a work about some work about iPS cells and iPS cell-derived myogenic progenitors.
Prepare like that. Okay. So first of all, I would like to thank all the organizers for giving me the opportunity to give you this talk, and let me bring you the apologies of my supervisor, Dr. Tedesco, which cannot be here today due to last-minute clinical commitments. So today we'll show our recently published data about the transplantation of iPS cells-derived myogenic progenitors in limb-girdle muscular dystrophy 2D model. Okay, so I don't think I have to introduce muscular dystrophies to this audience. What you might know is that there are a lot of preclinical and clinical studies which are ongoing to treat muscular dystrophies, and our lab focused its attention on gene therapy using stem cells. Okay, the stem cells we used was mesoangioblasts, which are well characterized in the lab, and are vessel-associated progenitor cells, which express the alkaline phosphatase and which are pericyte-derived and are present in the adult skeletal muscle of the patients. These cells has been shown able to contribute to the muscle regeneration in mice and in dogs, and are suitable for intraarterial delivery into the circulation. With these cells, there's a clinical trial, a phase 1/2 clinical trial ongoing here in Milan at Ospedale San Raffaele about allogeneic transplantation of mesoangioblast in Duchenne muscular dystrophy patients. So we took advantage of data previously published from the lab about transplantation of murine mesoangioblast in sarcoglycan null mice. The sarcoglycan null mice is the model for limb-girdle muscular dystrophy 2D. And in order to translate this strategy to human. Unfortunately, there are no limb-girdle muscular dystrophy large animal models, so we had to generate a novel immune-deficient sarcoglycan null mouse to act as a recipient for our cells. And then we attempted to isolate human mesoangioblast from limb-girdle muscular D2D samples. Unfortunately, we weren't able to isolate the cells, so we weren't able to isolate alkaline phosphatase pericytes, both from biopsies or banked cells derived from limb-girdle muscular dystrophy 2D patients. And whereas we were able to obtain cells which was alkaline phosphatase, these cells was functionally defective, so they wasn't differentiating, and they had a reduced proliferative ability. So we made one step back just to see if to see if we could confirm this data, and we analyzed biopsy obtained from patients with limb-girdle muscular dystrophy 2D patients, and we have realized that the number of alkaline phosphatase progenitors present in the muscle was significantly reduced. We analyzed also biopsies from the sarcoglycan null mouse, and we have seen that the result is similar. So, okay, I don't know what's happening. Okay, I'll try to explain it. Okay, basically, we started from cells derived from the patients, from banked cells or cells that we have derived from the biopsies. With the Yamanaka's factor, we reprogrammed the cells to the iPS cells that Massimiliano has introduced before. And then we induced a mesodermal commitment from the iPS cells to mesoangioblast. In this case, we named these cells human iPS-derived mesoangioblast HIDEMs. Then we genetically corrected these cells with a lentivector containing the alpha-sarcoglycan gene, which was missing in the patients. And then we transplanted these cells into sarcoglycan null mice, immune deficient. Okay, the first step was to analyze the cells for problems that could have been occurred with the differentiation protocol, and we have seen that the karyotype is normal. The reprogramming factor that was used to induce the reprogramming to iPS was silenced. The proliferation ability of the cells was comparable to the one of adult mesoangioblast. And then we analyzed the global expression profile of the cells, and we have concluded that the HIDEMs clusterized really close to adult mesoangioblast. So these will resemble the expression profile of the mesoangioblast. On top of this, we have made tumorigenic assays with these cells, and we never observed tumors in the mice transplanted with these cells. Then we analyze the surface marker of our cells just to make a comparison between adult mesoangioblasts and the hydens for the surface marker that we normally use to characterize mesoangioblasts. Moreover, we concentrated our attention on CD56, which is negative in mesoangioblasts since it's a marker of satellite cells, to the alkaline phosphatase, and to CD146, which is a perithelial marker. Okay, so we wanted to know if our cells could be able to differentiate in vivo and to generate myotubes in vitro. In order to differentiate these cells in vitro, we infected the cells with a vector containing the MyoDER cassette, which has been developed by one of our collaborators. And as you may see in the image, there's a widespread presence of hypertrophic myotubes that are labeled here with myosin heavy chain, which is a marker of terminal differentiation. So then we tried to translate this strategy and to transplant the cells. In order to transplant the cells, we have generated a muscle-specific sarcoglycan lenti-vector to correct the genetic defect of the cells, and the sarcoglycan null mice has been crossed with a SCID-BEJ immune-deficient mice. This to allow the allogeneic transplantation, the interspecific transplantation between human and mouse. Upon intramuscular injection, we were able to observe after 1 month from the transplantation the presence of large clusters of sarcoglycan-positive fibers, which colocalize with the human cells, which are labeled here in green with lamin AC, which is a marker of the human nuclear laminar. We then transplanted our cells intraarterially to see whether these cells resemble the mesoangioblast behavior. And after 12 hours, you can clearly see one human nuclei outside from the vessel, which in this image are labeled in red with CD31, which is a marker of the vessels. We made just one step back to see if the efficacy of engraftment of our cells upon intraarterial transplantation was comparable to the one of the cells straightly derived from the patient without the reprogramming. And we have seen a positive level of engraftment of our cell compared to the cells directly derived from the patient. After 1 month from the transplantation, we were still able to see alpha-sarcoglycan positive fibers colocalized with human nuclei. So in order to see if, to extend this strategy and to see if this is functional, we made another step back and generated iPS from murine cells, and then we reprogrammed murine iPS to MEDEMs, so murine iPS derived mesoangioblast. We transplanted these cells into the very same mouse, the sarcoglycan null mouse, after labeling the cells with GFP, and we have seen large cluster of GFP positive fibers, which were also sarcoglycan positive in the muscle tissues. The animals has been also tested for functional amelioration, and we have seen a functional amelioration upon the treadmill test, and the muscle explanted from the animals has shown an increased force measurement, an increased force at the measurement, both at the muscle level and a single fiber level. We analyzed the tissues also to discover if the alkaline phosphatase niche has been reestablished with the cells that we have transplanted. And as you can see, this is one of the sections of the muscle that we have transplanted. You can see the colocalization of the alkaline phosphatase with a green cell. Okay, and so we have counted also the total amount of alkaline phosphatase cells in the samples that we took from the mice, and we have seen an increase of the total number of alkaline phosphatase cells present in the treated animals compared to the untreated one. So in conclusion, we have shown a reduction of the alkaline phosphatase pericytes in limb-girdle muscular dystrophy 2D patient. We have shown that it's feasible to derivate pericytes from limb-girdle muscular dystrophy 2D iPS cells. We have shown the engraftment of these cells in sarcoglycan null SCID-BEJ mice, also through the through the intraarterial transplantation. On top of this, we have shown that the method is functional, at least in an intraspecific transplantation setup. Future plans from the lab includes the translation of this approach to other forms of muscular dystrophy, such as Duchenne muscular dystrophy, and to other platforms of genetic correction, such as human artificial chromosomes that we have previously used to correct the genetic defect in cells from MDX mice, from Duchenne muscular dystrophy model mice. We are also working on disease modeling and drug screening and tissue engineering using these cells. So most of the experiment that I showed you has been done in collaboration from 2PI, Giulio Cossu and Francesco Saverio Tedesco, which is now establishing his new team at UCL. I would like to acknowledge the people involved in the project: Sara Maffioletti, Martina Ragazzi, Sara Benedetti, and Yotoshi Shia, all the members of the lab, the long list of our collaborators, and our funding agency. All of you for your attention, and I'm happy to take your questions.
About the efficiency to cross the wall of the vessel when you want to transfect a whole muscle. I think it is still easier just practically in a mouse than in a human being. Do you anticipate any problems with just the transport of the progenitor cells?
Okay, about the transfer of the mesoangioblast through the interarterial circulation, there's a phase 1/2 clinical trial ongoing at San Raffaele Scientific Institute here in Milan, with 5 patients treated intraarterially with mesoangioblast in an allogeneic setup. So it seems to be feasible to transplant these cells. No major complication appeared.
Domanda abbastanza semplice.
A very simple question. You have shown a whole list of different muscle dystrophies that might benefit, or actually which you are thinking about working on. Are there any evidences compared to what we know, different mechanisms between the different dystrophies, intracellular mechanisms that might let you think that this type of a method might be more functional, might be better applied to one dystrophy type? Well, in our case, particularly as regards 2D, the fact that the stem cell pool is within the muscle, well, that might turn this therapy into a valid alternative to the examples shown by Professor Mendel before. Fundamentally, everything depends on the type of genetic correction which is used for the defect. One of the girls in the laboratory is working on empathies, deriving the items from patients with this kind of mutation. Each single pathology, of course, will have the own system. Fundamentally, this is a method to obtain progenitors that may be then transplanted. In your process of differentiation, I wanted to know if you are investigating the switch point between when the mesoderm genes are induced and the following phase. Well, fundamentally, the protocol simply consists in a media switch with growth factors towards the mesoderm. Let's get back a little bit. Fundamentally, what we investigated is, first of all, for these markers, the classical markers we are using, these are in terminal state? No, this is the situation. They are the starting IPS. This is the HIDEMS situation exactly in the middle of the protocol, so we stop the culture. So the HIDEMS, sorry, human, this is the definition of HIDEMS, of course, as I think you know. This is the situation mid of the protocol. 3 weeks, more or less, yes, starting from the IPS colony to reach this situation, about 3 weeks and a half. What is still very time consuming is the IPS generation that actually requires time. So the point is that a question appears not only on the muscle lineage, but also as regards to the differentiation and the other turning points in the process. I wanted to know whether in generating these cells you can actually find any pathway which is similar to satellite cells. Well, actually, we considered this fact because we had a doubt whether promiscuity might exist between the two lines, and in some cases we actually tried to make some faxes on cells about to differentiate, so after the injection, and you can see a small percentage of cells activating C56, so there might be a sort of myogenic memory. Sorry, we cannot hear if he doesn't speak in the microphone. Well, based on the data we have published, all the cells we have differentiated from IPS once induced with MyoD, well, the difference is not, is like in the picture I have shown.